It's Rory Kirchner. I work at the Harvard School of Public Health and the Bioinformatics Core. And I'm going to talk about how we handle our most common requests, which is to do RNA-seq differential expression analysis, and talk a little bit about how we validate our process for handling that type of data. So if you search Google for RNA-seq analysis pipeline, this is crazy, but there's almost 500,000 hits. But there was a post on the RNA-seq blog. There was a poll, and the poll question was, what's the biggest obstruction that's keeping you from doing your RNA-seq analysis? And the number one result of the super deep poll of about 150 people was that there's a lack of a standardized data analysis pipeline. And the second most common missing piece was that there are a lack of skilled bioinformatics specialists to run the pipeline. So there's a need for something that's reproducible and something that the average person, an unskilled person, can download and install and get going to do their work. So the reason why there's a need for these type of pipelines, if you do any kind of NGS analysis, there's 50 different pipelines for every different type of analysis, is that handling this data is super complex. So, and the complexity for the researchers starts even when they are first trying to install the tools that they want to be able to do their analysis in the first place. So if you are in a weird compute environment and you have, you're on some legacy system where you have all kinds of stuff injected into your path, it might be hard to even build the libraries to build the tools to do what you need. And so that's a roadblock for a lot of researchers. If there's also, for each step of any sort of analysis you want to do, there are many choices for the tools that you can use. So for example, there's maybe 30 different splice read aligners, and all of them, if you read the papers, they all claim that they do the best. And so you have to choose what tools you want to use and then learn how to use each tool. Handling the data itself is complex. So if you look on seq answers or biostars, a really common question is, why don't my annotations match? And then finally, even if you could install the tools and you had all the data in the right format, tools that output a BAM file, for example, don't always output the same BAM file. So on the right is hard to see, but these are the things that need to be correct about your BAM file in order to run the Broad's RNA-seq QC tool. So there's a bunch of different steps that an aligner or whoever made your BAM file, whatever tool made your BAM file, may not set correctly. And so to get tools to work together, there needs to be a lot of glue code to make them work together. So that's why there's so many of these pipelines. So we, at the School of Public Health, we have a system for implementing NGS analysis pipelines called BC Bio NextGen, and Brad has laid out some development goals of BC Bio NextGen, which I think bear keeping in mind all the time. So we do, we work on GitHub, and we work with a bunch of different researchers, both in academia and in industry. So multiple people are working on the pipelines, multiple people run them in production, and so if there's regressions, we pick them up that way. If there's new features that want to get added, they get added constantly. The other goal is that the analysis should be scalable and easy to install. So you should be able to start from a bare machine, install everything you need to run an analysis that you can run on your laptop and to run some validation data. And then if you need to scale up to do your whole analysis, it should be just as easy to deploy it on your big compute infrastructure and run the same analysis. And our stuff is well documented. We have a great read the doc site with examples and test data that you can use. And the last important bit is that's quantifiable. So at the end of an analysis, roughly how you did, and that way we can test for regressions when we're updating tools or changing the way the analysis is done. Just briefly, to handle the installation, we use Cloud by Linux to do a lot of the tool installs, in addition to some package managers for Python and there's a package manager called Homebrew for OS X that has some nice simple Ruby formula that is also available for Linux. And the good thing about this is you can install all the tools without sudo. So a lot of times on a cluster environment, you want to try out a new tool, you have to go bug the system to install the tool for you. But all of this will install in your own directory or whatever you want without sudo. So our system is pretty easy to use. So the tools are pre-configured in that we try to learn as much of the options that you should set for each tool from the data itself, and that requires the researcher only set a couple high-level options of the type of analysis they want to run and to describe their samples. So the analysis involves just sticking your FASTQ or BAM files in a directory, create a metadata file that describes the samples, and editing a small configuration file that just says what genome you want to run and what type of, how your library was made and we set other parameters for the tools as you go. So this is just the standard RNA-seq analysis pipeline. You provide the either pre-aligned FASTQ files or BAM files or FASTQ files. We do some QC on the raw data, run alignments with top hat or star. We also do sailfish for alignment-free work. We do some quality control on the resulting alignments, do some quantitation of the transcripts, and I'll put a report that a researcher can look at so you can spot any outliers or any problems with your data. We also do some transcript assembly and try to filter out some bad assembly from that data. And then at the end, we do differential expression where we run a bunch of different differential expression callers. And if we automatically detect if you have ERCC spike-ins and do ERCC concordance. If your data set happens to be from the CompCoder data set, which is from the FDA, it's two known reference samples that you can test differential expression callers against. So if your data set is from the CompCoder data set, it will run a concordance test with all the callers. And then at the end, I'll put a bunch of different caller comparisons. So it's important to do the last step where you check callers against each other because there's varying differential expression calls across all the methods. So this is from a nice paper by Charlotte Sonneson. She wrote this R package called CompCoder that does all the count-wise comparisons. And you can see that there's quite a bit of variability between what's differentially expressed depending on what caller you use. But the CompCoder data set isn't the best tool to do validation. So because it's technical replicates, so we also provide options to do simulation of reads. So right now we just provide count-based simulation to just check the downstream callers, but we have some scripts to generate simulated FASTQ data from using the Flux simulator that we'll push in. But that lets you do kind of more rapid testing of all the count-based callers. So having a pipeline set up that we can validate at the end lets us answer some questions. For example, there's a big discussion on the blogosphere between Mac McManus and a couple authors of a paper. The authors of the paper claim that we should trim, quality trim RNA-seq data, and we should trim it as aggressively as possible because that allows you the highest percentage of reads that align. But the counterpoint that Matt was making is that the cost is that you have less reads that align total. So there's some back and forth on the blogs about which notion was correct, but there was no conclusion about whether or not it affects differential expression at all. So we can take a first check at that by flipping where we trim at, run the pipeline, and just checking on the CompCoder data, and then checking how we do against calling with the CompCoder data, and the results are virtually identical. So, but the CompCoder data happens to be like a very high quality data set. But this is like an example of the kind of questions that you can look at with having some kind of validation data set to work with. So using simulated data, this is a figure I like to send to our potential collaborators. So a lot of times you have the question people want to know is how deep should we sequence? And so this is an example of, on the left, if you were to run 3 replicates at 100 million reads, and the graphs are faceted by the log2 fold change, and each bucket should go 200, 400, 600, 800, 1,000 if you could get all the reads. So at 3 replicates, you can see that you don't get all the 2-fold changes, and you kind of top out. You don't do it much better at 1.5-fold changes. But if you have the same number of reads and you do 15 replicates instead of 3, you can get almost all the 2-fold changes and a good amount of the 1.5-fold changes. So this is another example of the usefulness of having something that you can plug into your analysis pipeline and generate some results on simulated data. We do all our development on GitHub. You can install it. The install script is just grabbing this file and running it. We welcome contributions, and so thank you.
Any questions? So, you get Mike? Sure.
So I'm curious, the install script here, that's for installing it on a single node. Do you know what the approach would be to install this over a cluster? Yeah, so we kind of assume that you have a shared file system, so you'd install it on the one and replicate it across, or else you'd have to install it on all the nodes individually.
For the differential expression step, what tools do you use, or have you implemented your own?
We use we use count-based tools usually. So, and the tool that we use the most is RNA-seq. All right, thanks.
Thank you. All right, thank you.